Hi, everybody. Today we're going to talk about Chapter 1. And Chapter 1 is just a really basic overview of microbiology. For every chapter, I like to break it down into certain topics. This makes it easier for us and we don't get overwhelmed. So, in Chapter 1, there's 5 main topics we have to talk about. We're going to talk about the branches of microbiology, different types of microbes, disease, specifically infectious disease, then the boring part, the founders of microbiology, and then lastly, how we name and classify microbes. So in this first video, I'm going to go through 1 and 2, branches and the different types of microbes. Whenever I ask students what microbiology is, they'll say something like the study of bacteria or the study of stuff that's too small to see with the naked eye. You need a microscope. That's not necessarily true. So microbiology is a study of all microorganisms. That does include bacteria and viruses. Both of these are really, really small things. Bacteria you could see with a regular compound microscope, like in the lab, but viruses are so small and they're not even alive. They're non living parasites. And viruses are so small, the only way to see them is with an electron microscope. And it's very hard to find an electron microscope because they cost anywhere from $1 to $2 million. So that's to tell you how small viruses are. But then there are also microorganisms, the eukaryotic microbes, that are actually big enough that you can see them with the naked eye. So, for example, fungi such as molds, mushrooms, right? You could definitely see fungus. These worms, parasitic worms, these worms are called helminths. Helminths include things like roundworms, flatworms, tapeworms, things like that. Parasites, right? The other main microbes include the protozoa and the algae, and we're going to talk about all of these in more detail in a bit. But first, let's talk about some of the different fields of microbiology. A lot of people think that a microbiologist is just this boring scientist who's just working in the lab all day and looking under a microscope for like 12 hours a day. But that's not true. I mean, I'm a microbiologist and I barely ever looked at a microscope once for my whole PhD because there is so many branches of microbiology. So the first major obvious branch of microbiology is public health microbiology or epidemiology. So these are individuals like the famous Dr. Fauci, right? These are individuals that study the spread of disease. They're looking to track and predict where diseases are going. What is it going to look like months from now? Who's at risk? Who is most susceptible to certain diseases? Can we predict based on climate change or based on travel how diseases are going to spread in a population? So, public health is one of the major fields of microbiology, and we will talk about it this semester. Another field is environmental microbiology. Because microbes are everywhere, they can shape everything about our planet. They're in the soil, they're in the water, they're in Antarctica. So, microbes really do change the soil. They affect things in the earth. One of the most important ways that we apply microbes to the environment is a process called bioremediation. Bioremediation is taking microbes, using microorganisms to clean the environment in some way. Because microorganisms don't just eat food like humans do, some microbes can eat aromatic compounds, toxic compounds that would be toxic to humans. Let me give you an example. About 10 years ago, there was this major oil spill. And one of the ways that we actually cleaned up the oil that was contaminating the ocean was to actually seed the area of contamination with microorganisms that could eat the oil and break it down into harmless substances. So, bioremediation is a really important application in microbiology. Another field is biotechnology. So, this is when we take microorganisms and we use them to produce really valuable products that we want and that we could sell and make a lot of money on. So, for example, I'm sure you know that beer, wine, alcohol, things like that are fermentation products of yeast or bacteria. We can actually take the waste products of these microbes and produce stuff we like the beer. Vaccines are also produced by microbes. Not only are vaccines made to prevent disease, we actually produce them using microorganisms. I'm sure you could think of many other products that we use microbes to make, such as yogurt, cheese, bread, a lot of different things. Another field is immunology. A lot of people, when they think immunology, they think right away about vaccines because, yes, one of the major jobs of immunologists is to formulate and produce and develop vaccines. Other things immunologists do is they look at allergies. Allergies is a hot topic right now because we seem to have a lot more of it and it's emerging. So, immunologists will look at how our immune system responds to harmless things like allergens or maybe immunodeficiencies, where the immune system becomes weakened. For example, the HIV virus, which can weaken the immune system over time. Another area of immunology is the study of autoimmune disorders, where the own immune system starts mistakenly targeting an individual's own cells. Immunologists do so many things now, and it's going to be a really hot topic in this class. Something that is really important to me, because this is something I do in my field of research, is genetic engineering. Now, this is where we take genes from one organism and put them into another. We're creating a recombinant microorganism. So, for example, we could take something very basic like E. coli, and we could pop a human insulin gene into the E. coli bacteria, and then we could use the bacteria to produce human insulin. This is actually how insulin is produced. The insulin that diabetics use today is mass produced by bacteria. It's very cheap, it's very cost effective. We could grow E. coli very quickly in the lab. And because microorganisms are cells, and they do a lot of the same general physiology that humans do, we can actually study microorganisms and apply some of these, what we learn, to humans. So that's one reason why microbiology is such a major field today. Agricultural microbiology. One of the things we have to worry about is because there's so many humans on this planet, is producing enough food for everybody. And because fungi and bacteria, they can cause not only human disease, but they can also cause plant disease. So we really need to make sure we prevent disease to our crops, like to our corn, to our tobacco, to our wheat, to our bananas, you name it. We need to try to prevent the spread of disease to our crops so that we don't all starve. Food microbiologists. In this class, we're going to talk a lot about food microbiology. There's three main areas in food microbiology that we're going to talk about. Obviously, microbes can be used to produce food. Like we talked about, cheeses, yogurts, bread, beer. All right, so we can use microbes in food production. Second thing we have to think about is food spoilage. What are some things we can add to our food to help prevent them from spoiling? I'm sure you could think of some like salt, acid, preservatives, things like that, because we don't want our food to spoil so fast before we can get it from the grocery store to our house, right? So we actually need to come up with these kind of preservatives. And then the third thing we got to think about are foodborne illnesses. I mean, almost every year something pops up in the news. Most recently, there was a lot of E. coli outbreaks in romaine lettuce. You probably remember the lettuce department was like empty because of this E. coli spread. So we always have to be concerned and thinking about how can we prevent foodborne illnesses. So these are things that are going to come up a lot this semester. Now, lastly, I'm a big fan of these tables because they're really helpful. I'm a big table person. This one's pretty basic. This one just tells you the branches of microbiology. So some of them are pretty obvious, like bacteriology is the study of bacteria, duh. Virology, the study of viruses. The one that's not so obvious is this one, mycology. Anytime you see this prefix myco, or I should say most of the time, this indicates something to do with fungus. So mycology is the study of fungi. If you see mycosis, this means a fungal disease. So except for the one or two exceptions that I'll tell you about later, myco indicates fungus. Now, in my PowerPoints, I put these little review questions. These are good for you to go through on your own. And when you have a question or you don't know one of them, send me an email and we can make sure that you get on the right track. When we think about microorganisms, we really classify them into three main groups. Okay, so the living microorganisms, they are either prokaryotes or eukaryotes. And then the non-living microorganisms, such as viruses, they are what we're going to call acellular parasites. So remember, I always like to make a table. I'm a table person. So let's make a table comparing these three groups of microorganisms. So we have the prokaryotes, the eukaryotes, and then what I'm going to call the non-living infectious agents. So let's compare these things. So prokaryotes, these are the earliest forms of life. They're usually also the smallest cells. They're very small cells. Eukaryotes, on the other hand, these are more recently evolved. Eukaryotes are going to have larger and more advanced cells. Now, the prokaryotes, what makes them unique is that they do not have a nucleus, whereas eukaryotes, they always do have a nucleus. Remember, the nucleus is where the genetic material is stored. It's the location of the DNA and the genetic material in an organism. Because prokaryotes do not have a nucleus, their genetic material is actually just kind of tangled up inside the cell. It's kind of like bunched up. So the DNA is bunched up within the cell, but there's no membrane bound nucleus protecting it. That's the major difference. Another major difference, prokaryotes are always single celled, which means there's just one cell. That's it. Whereas eukaryotes can be either single celled or multicellular. And I'll give you some examples of both. The last major difference is prokaryotes do not have any membrane bound organelles, whereas eukaryotes have the membrane bound organelles. Membrane bound organelles include those weird things that you learned about in anatomy and physiology, like endoplasmic reticulum, the Golgi apparatus, lysosomes, the vesicles, things like that. Those are found in eukaryotic cells, but not prokaryotes. A lot of people think that ribosomes are membrane bound, but they are not. Let's put a little star here so we don't forget. All cells have ribosomes. So even prokaryotes have ribosomes. That's really important because ribosomes contain a special kind of RNA called rRNA, which stands for ribosomal RNA. Ribosomal RNA is really important in classifying cell types. So we're going to remember that one for later. Now let's talk about those non-living infectious agents, okay? What makes these guys different? First of all, these are not cells. They do not contain any cells at all. They're not cells, meaning they're not living. They're not alive. Secondly, because they're not alive, that means they need a host in order to replicate. If something needs a host to replicate, we say it's an obligate intracellular parasite. Obligate means it needs, intracellular means in the cell. So these things need to get in the cell. They're parasites. So we are not going to confuse these non-living infectious agents with living cells, prokaryotes or eukaryotes. Now that we have gone through and described the three main classes of microorganisms, either prokaryote, eukaryote, or non-living, there's something else really important we need to know about classification. And this is the three domains of life. Whenever we're classifying an organism, the highest level of classification is the domain. And there's only three, and you need to know these three. Okay, so the first major domain is the bacteria. They are their own separate domain of life because they're very unique. The second major domain of life is called archaea, and the third is called eukarya. What makes all of these three different domains unique? Remember I told you that ribosomal RNA, all three of these domains, we know that they are very different because they all have very different ribosomal RNA. So rRNA is what's used to classify an organism and fit it into its proper domain. So if you looked, for example, in eukarya, this includes a lot of different things like humans, we are eukarya, animals, plants, fungi, those worms I told you about, the helmet, algae. Even though all of these groups are very different, we all have the same structure of ribosomal RNA. So that puts all of us into the eukarya domain. Bacteria and archaea, though, their ribosomal RNA is significantly different from us and from each other. So that fits them into their own domain. Okay, so those are the three you just need to remember. As I'm sure you could tell, eukarya have eukaryotic cells. When you go back to our table, all of these things in eukarya, we all have nuclei, we all have membrane-bound organelles. Bacteria and archaea, on the other hand, these are both prokaryotic cells. So bacteria are prokaryotic and archaea. They both are single-celled organisms without a nucleus and without membrane-bound organelles. Now that we know the domains of life and the three main groups of microbes, we have yet another table to fill out. I told you I love tables. This table shows you every single group of microbe, and yes, we're going to study every single one of them this semester. And we're just going to go through some basic features of each. The first microbe type is the bacteria. Remember, bacteria are in their own domain, bacteria. What type of cell do they have? They have single cells, and they are prokaryotes. Key traits about bacteria. Guess where we find bacteria? We find them everywhere. They're in your body, they're on your skin, they're in your refrigerator, they're on your doorknob, they're in the ocean, they're in the soil, they're in your dog's mouth, okay? They're everywhere. The other important thing we need to know about bacteria is they have a cell wall, and we're going to talk a lot about this cell wall in week 2. What's so unique about bacteria's cell wall is it contains a material called peptidoglycan. I'm going to put this word in like a bright pink color so you don't forget it. If there's one thing you're going to learn today, it's that peptidoglycan is in bacteria. It's in the cell wall of bacteria. Peptidoglycan, say it 10 times out loud right now. This is going to be your new best friend this semester. If you learned one thing this whole semester, it's going to be about peptidoglycan. It's so important. So be ready to learn all about it. Our next microtype is the archaea. So archaea, remember, they have their own domain. They are also in the domain archaea. So they're unique. Archaea have single cells. They are, as we said, prokaryotes. Now, key trait about archaea. Archaea are not found everywhere like bacteria are. Archaea tend to live in extreme environments. They are what we call extremophiles. Extremophiles mean they love extreme environments. So think about some extreme environments such as Antarctica, a volcano, a hydrothermal vent, a hot spring, things like that. That's where you're going to find archaea. Now, archaea, they do have a cell wall, but what makes them different from bacteria is there's no peptidoglycan in their cell walls. We will talk a little bit about archaea this semester, but because they're not really medical pathogens, we don't talk about them too much. We'll just talk a little bit about them. Our next group is the protozoa. So protozoa are in the eukarya domain. However, these ones are single-celled. So even though they're eukaryotic cells, they don't have many cells. They have one cell type, they're single celled. But they have eukaryotic cells, meaning they have nucleus, they have membrane-bound organelles. Key traits about protozoa is they can be free-living, but many of them could be pathogenic. They can be parasitic, and they can cause disease. I'll give you an example. I'm sure you've heard of malaria. Malaria is a disease caused by a protozoa. Maybe you've heard of toxoplasmosis. It's associated with cats. It is also a protozoal disease. There's also a common STD. It's called trich, also protozoa. So we will talk a lot about protozoa this semester because they cause some of the most abundant diseases out there. Our next group is the algae. Algae are in the domain eukarya. However, algae could be single or multicellular. There's many different types of algae, and they all have eukaryotic cells. Key traits of algae. I'm sure you know, algae are photosynthetic. That means they gain energy from sun. And yes, we will talk about algae this semester. Believe it or not, there is a toxin producing algae out there. It's associated with what we call red tides, and it's an algae that releases a neurotoxin. So, yeah, we'll talk about algae for sure. The fungi, they are eukarya. Fungi can be single celled, it's usually a yeast, or they could be multicellular. We call it a mold. So, fungi are eukaryotic. They have nuclei, they have membrane bound organelles. They are mostly decomposers, meaning they eat dead and decaying matter in the earth. Fungi and bacteria are both types of decomposers. And if this organism obtains its energy from eating dead, decaying matter, we typically call it a saprobe. So, saprobe is a fancy word for eating decaying matter. Now, as you know, fungi can affect humans. They're typically what we call dermatophytes. That means they can cause skin, hair, and nail diseases. I'm sure you've heard of like athlete's foot and things like that. So that's fungi. And there are some diseases that fungi can cause internally as well. The helminths, we remember these ones are the parasitic worms. Helminths are also eukarya. They are always multicellular though, and they're eukaryotic cells. So what are some key traits we need to know about helminths? Well, what we need to know is they are parasitic, so usually they need some sort of host and supportive at some point in their life to thrive. That could include a human, an animal, an insect, or a fish, or something like that. Helminths all have primitive organ systems. So there's some of the earliest life, the earliest multicellular life on Earth. Now, our last three here, these are all the infectious agents, so non living. So let's talk about viruses. We know viruses, these are not cells, these are infectious agents. They're not alive. So that goes for viruses, viroids, and prions. These are all not cells. They're all those infectious agents. Now, traits about viruses. Viruses are made up of two things. They have to have some sort of genetic material. So that genetic material could be either DNA or RNA, but never both. So, for example, coronavirus is an RNA virus. Influenza is an RNA virus. They all only have RNA, whereas herpes is a DNA virus. The HPV virus is DNA. It doesn't have RNA. So every virus is different and it has one or the other. The second thing all viruses have is something called a capsid. This is a protein coat that surrounds the genetic material. So for now, those are the two things you need to know about viruses. They all either have DNA or RNA, and they all have to have that capsid. Now, viroids are different than viruses because viroids contain only RNA. They are simply just infectious RNA. Viroids do not have any capsid or protein at all. They're just RNA alone. So there's no protein in a viroid. As far as we know, all of the viroids that we know about infect plants only. So all the ones we know about infect plants. There could be some out there that infect humans or animals, but we haven't discovered them yet. Because they're so small and they're just RNA, it'd be really hard to find them. Our last infectious agent are the prions. And prions are infectious protein. They contain no genetic material at all. If there's one thing that scares the daylight out of me, it's the prions. It's beyond strange to me. How could this infectious protein spread? Well, I'll tell you. It spreads by ingestion of contaminated meat. Usually, it has to be contaminated neural tissue. So, for example, mad cow disease. I'm sure you've heard of it. It spreads from eating infected cattle, like brain or neural tissue that has the prion in it. So, for the most part, prions only are spread through eating the meat. There's very, very few cases, but it has been heard of, that a prion was on a medical equipment and there was like a brain surgery or something, and the prion is introduced into the brain through the equipment that wasn't sterilized properly. But that's very, very rare. In fact, prions in general are extremely rare, so don't freak out and, go vegetarian or anything like that. So, that is my table. of all of the types of microorganisms, and we will be studying all of them this semester. And here is again the review to look over.